Okay, so here we are on another snow day, and I hope that you guys are all warm and dry at home. We're going to carry on with chapter 4, and you will all hopefully review this lecture before we have class on Monday, to sort of just fill in this lecture gap. And then this lab that we were supposed to do today will just occur next Wednesday. Okay, we'll keep pushing it out. We've got about to use up the last of our empty lab spaces. So if we have any more missed labs, we'll have to use our reserved makeup day, which I believe is on advising day. So we left off talking about the endomembrane system and talking about how proteins are modified as they move through the endomembrane system. And we looked at this example here, which looks at the formation of a immune system protein, this immunoglobulin here, that's going to go out and leave the cell to do some work. I want to review that with another type of protein. Here we again are going to start down at the nucleus, and just outside of the nucleus, we have the rough endoplasmic reticulum, right? And that rough endoplasmic reticulum is studded with ribosomes, rough with ribosomes. And those ribosomes are producing the proteins, and the proteins are moving into the interior lumen here of the endoplasmic reticulum. So as that protein is formed, it's going to move through the endoplasmic reticulum and it's going to be modified as it goes. Remember, when our protein first starts out, it has primary structure, and it might, as it moves through there, be folded into secondary or tertiary structure. It might have some groups added to it to change its chemical composition or to change its structure, right? So in this example here, we're making some digestive enzymes. And so these digestive enzymes are synthesized on those ribosomes, and they are modified as they travel through the rough endoplasmic reticulum. Ultimately, they're going to be packaged in a vesicle that is going to leave the endoplasmic reticulum and travel over to the Golgi apparatus. When the vesicle comes in contact with the Golgi apparatus, it can fuse. Remember that membrane is a very fluid substance, and so it's going to fuse with the Golgi apparatus, and those protein contents are going to move into the Golgi. As they move through the Golgi apparatus, they're going to be further modified. They're going to start to take their final shape and become, in this case, these functioning digestive enzymes. And they'll be packaged up in lysosomes to move away from the Golgi apparatus. So, what's this term lysosome? Lysosome is essentially just a specialized vesicle. So, it's a membrane bound bubble, right? Just a little membrane sac. But inside that lysosome are digestive enzymes. And so the term lysosome only applies to vesicles that are carrying digestive enzymes. So these digestive enzymes, remember we talked about the importance of that compartment when something is kind of dangerous, right? Because those digestive enzymes can digest things inside the cell, not just things that the cell wants to eat, but it could actually digest its own structures if it's not careful. So we package those digestive enzymes into this lysosome, and that lysosome will actually break down food material that comes in from outside the cell. So if we look in the interstitial fluid in this diagram, we have some food particles that have traveled from the digestive system. They've moved through the body and the bloodstream, and they've arrived at the surface of this cell. And so, because of the fluidity of that membrane, as those food particles gather on the cell, they're going to form another vesicle, and that vesicle is going to move into the cell. Now, this particular vesicle is called a food vacuole. And again, just a fancy name we're giving to the same basic structure, a little membrane bound bubble that contains food. So the food vacuole is going to move in. Maybe it's got some carbohydrates, some tasty sugars in there, and it's going to fuse with our lysosome that was coming out from the endomembrane system. When these two vesicles fuse, their membranes bind together and they just surround all of those contents as one unit. So the vesicle becomes bigger, right? And now those digestive enzymes can be used to break down that food. So remember, we talked about a hydrolysis reaction, right? When this happens, our little macromolecules that have traveled to the surface of the cell are going to be hydrolyzed into their monomers. They can be released into the cell to be used as building blocks for different molecules, or they can be used to produce energy. Okay, so now I want to move on from the endomembrane system and talk about the rest of the organelles. And so we'll first start with an organelle that only exists in plants. And so this is a structure that's specific and unique to plants, and it's called the central vacuole. And the central vacuole is kind of just a big reservoir that can be found usually pretty centrally in the cell. And if we look over here, this entire space, big empty opening of white, is all the central vacuole. Remember we said plant cells could be up to 90% water? A big portion of that water is actually just stored as fluid water in the central vacuole. Other things that can be stored in the central vacuole include waste products and toxins, um, additional pigments that can be used for photosynthesis, and the importance of the central vacuole, um, in a lot of ways for the plant cell, is actually to provide structure. And so at the end of this chapter, we're going to talk a lot about the movement of material into and out of membranes, and we're going to talk about osmosis and diffusion. And we'll look at the role that the structure plays in the plant cell. But basically, what happens is when the central vacuole is full, I'm struggling with my pen here. When this vacuole is full, it causes pressure on the internal contents of the cell. And so as it pushes those contents out towards the plasma membrane and ultimately towards the cell wall, it pressurizes the inside of the cell. That makes the cell rigid or stiff, and that gives it structure. So when a leaf is, your houseplant leaf looks really wilty and collapsed, that's because the central vacuoles of that leaf cells are not full, and so the leaf itself begins to kind of fold in and kind of collapse on itself. And you water your house plant, and those leaves perk up and they stand straight up again, right? That's because their central vacuoles are full of water. Plants can also use the central vacuole as a dumping ground for their waste materials. And so if they produce toxins as part of their metabolic process, they can put them in that central vacuole and store them up. And some plants intentionally concentrate toxins in their central vacuole so that when a predator comes along, like an herbivore comes and takes a munch out of this leaf, it gets a mouthful of those toxic compounds. And it can make the plant taste bitter, make it unpleasant to eat, and it's a defense mechanism for the plant. So the central vacuole is sort of a multi-purposeful space, but it's a special sort of um, separate space within the central region of the cell. Now, some of our single-celled eukaryotes that are free-living beasts also have vacuoles, and these are a different type of vacuole. They're not called a central vacuole like in the plant cell. These vacuoles are actually used for maintaining water balance. So this organism over here is a protist. This is a single-celled eukaryotic organism that is free-swimming and lives in freshwater environments. And we'll talk again later in this chapter about why, but this individual um, organism is constantly taking on water from the surrounding environment. And because of that, it has to have a way to get rid of that water quickly in order to control its body and prevent its plasma membrane from bursting. So it has these specialized structures, which are shown here, and these are called contractile vacuoles. And basically, the contractile vacuole consists of collecting ducts that pull extra water from other parts of the cell and pull them into a small um, central reservoir. And as water moves into that reservoir, the vacuole itself will expand. It'll get bigger and bigger and bigger until it reaches its maximum size. And then the whole thing will squeeze and push all of the water from the vacuole out through a pore to the outside of the body. So it's kind of like constantly bailing out your body. And so this organism has a way to get rid of extra water. I'm trying to find, sorry. I was hoping that I could, I guess I'm going to have to escape. Um, so what I wanted to do was pull up this video that I have, and I'll post this video on our Moodle page so that you can look at it. But this video actually shows this contractile vacuole in action. And so we're going to look here, we'll play this. We're going to be watching this region right here. Can you see the vacuole filling? These are the collecting ducts, and they're pulling extra fluid. They're just squeezed and emptied. So now the collecting ducts dump their water into the contractile vacuole. A little more fluid is moving in and moving in, moving in. And as this vacuole expands and reaches its maximum size, it's going to squeeze again. And the collecting ducts are pooling water. They dump into the contractile vacuole. And so this is just sort of a background process that's happening in this animal's body at all times to try to rid it of excess water that's coming in from the surrounding environment. Okay. So those are vacuoles. Now let's move on and talk about the mitochondria. Probably you have at some point in time learned about the mitochondria. You might refer to it as the powerhouse of the cell, right? The reason that it has that nickname is because the mitochondria is going to be the organelle that produces most of the energy for the cell's work. Okay, it's going to produce ATP. And remember, we talked about ATP being really energy rich because of those negative phosphate groups being bound to each other. So when we break off a phosphate group, we release a bunch of energy. And so the mitochondria is going to carry out the process of cellular respiration. And cellular respiration is going to be the metabolic process that's going to produce lots and lots of ATP. And it's going to do this by taking the fuel molecules that are come in from the diet, mostly sugars and also the broken down products of lipids and proteins, and they're going to convert those into energy rich ATP. If we look at the structure of the mitochondria, it has a pretty unique and distinctive structure. It has both an outer membrane and an inner membrane. So here in the diagram, we can see the outer membrane sort of forming the outer boundary of the mitochondria. And then we have this inner membrane, and the inner membrane is folded intricately back and forth. And these folds are called cristae. And I know that if I asked you in class why we had all those folds, somebody would say that it is because it increases surface area, right? So this interior membrane has a lot of surface area on the inside of the mitochondria, okay? The outer membrane is sort of the protective outer barrier. You can see it over here in the scanning electron micrograph as well. And then the inner membrane is going to be selective and give us a lot of surface area for the reactions of cellular respiration to occur along. Okay. In the very interior of the mitochondria, you have the matrix, and this is actually a fluid-filled space. So we've got some more fluid in here. And inside this matrix, we also have the enzymes that are necessary to carry out cellular respiration. The mitochondria has its own DNA, so its own genetic material is in there. And then the mitochondria also has its own ribosomes. And all of those things are stored in that matrix. Now, mitochondria occur both in plants and in animals, right? We find them in our plant cells and in our animal cells. But the chloroplast, our next organelle, only occurs in plants. Remember, we talked about organisms that are autotrophs and can produce their own food. So they do that by using chloroplasts. The chloroplast is going to be the structure where photosynthesis occurs, and it has a similar shape and appearance to the mitochondria. And you guys hopefully all saw these chloroplasts last week when you looked at the Elodea leaves and you saw those green ovals that were pressed around the edges of the cell wall in our Elodea cells. Each one of those little green circles is a chloroplast. And this is where light energy is going to be converted into the energy of chemical bonds. And that chemical bond energy is going to be used to make sugars. And so we'll essentially be storing sunlight in sugars. So the chloroplast also has a structure that's somewhat similar to the mitochondria in that it has two membranes. There's an outer membrane, shown here, and then there's an inner membrane. The inner membrane actually does not have folds in it like our mitochondria membrane does. So the inner membrane is not providing surface area. Instead, the surface area of the inside of the chloroplast is provided by these little green disc-like structures that are shown here. And each one of those, sorry, each one of those little discs is known as a thylakoid. And each thylakoid is going to contain the pigments, the chlorophyll, that's essential for photosynthesis. And this is going to be where light energy is harvested. So the thylakoids are like the antennas that receive light energy. And the thylakoids are organized into little stacks. So here we have one thylakoid, and then right below it, we have another and another. And so we have a whole stack of thylakoids, kind of like we talked about the Golgi apparatus being a stack of membrane-bound structures. The thylakoids stack up so that, again, we have lots and lots of membrane surface area relative to the interior volume of the thylakoids. Okay. These stacks are called grana, granum plural or grana singular. And inside each one of these thylakoids, there's a separate membrane or separate fluid that is unique from the fluid that surrounds the stack itself, surrounds the grana. Okay. The fluid around the grana that is where all of these thylakoids are stacked up is known as the stroma. And the stroma also, like in the mitochondria, contains DNA and ribosomes. So our chloroplasts and our mitochondria both have their own independent DNA. That means that they can replicate themselves and make more mitochondria or more chloroplasts completely independently of what the cell is doing. And they can also use their ribosomes and that genetic information to make their own proteins and the enzymes that are needed to carry out their metabolic processes. So really the mitochondria and the chloroplast are unique structures within the cell and have an interesting potential origin. So we believe that both mitochondria and chloroplasts were originally free-living organisms. They were probably free-living prokaryotes, the chloroplast arising from a photosynthetic prokaryote, and the mitochondria arising from an oxygen metabolizing prokaryote. And it seems that these somehow were engulfed by a larger eukaryotic cell, probably as the cell was probably intending to use them as food. But once they were engulfed, if they didn't get digested right away and they continued to carry out their function, the cell may have realized that they were actually more beneficial if they kept them inside instead of eating them. So that would lead to what we call endosymbiosis. And endosymbiosis is this literally means living inside together. And so we have lots of examples of endosymbiosis now, both in microscopic life and in larger life, where one organism lives inside of another organism and the two organisms live together. If this situation is beneficial to both organisms, it becomes a mutualism. And with both organisms benefiting, in this case, we think that what happened is that they just ceased to be separate organisms, sort of like individual colonies benefiting from each other might cease to be separate individuals and become a multicellular organism. These mitochondria were so beneficial to the cells that had engulfed them in terms of their energy production that they would really want to keep them around. And the mitochondria are now safe. They're not going to be engulfed by any other cell or um, consumed by anything else because now they're inside this much larger cell. So this is our best hypothesis for where the mitochondria and the chloroplast came from, that they originated in some sort of mutualistic relationship with larger cells and ultimately came to be no longer independent living organisms, but instead an organelle with inside this larger cell. And we have some examples now where we have single-celled organisms like this organism pictured here is known as Hetina. And it has a very, very large chloroplast inside its body. It fills most of the volume of the organism's body. When the organism divides and becomes two new individuals, only one of them gets to keep the large chloroplast, and the other one immediately goes out and tries to engulf a free living algae cell to help meet its needs. And so this seems to be a case where this is maybe this process, evolutionary process is happening over again with this symbiosis. Be really fascinating to follow these guys and see if we can push them in the lab to quit being individuals. Okay, so now what I want to do is move on from talking about the structures, the independent organelles of the inside of the cell, and talk instead about the scaffolding and the girdering that gives the cell its overall shape and provides a lot of support and anchoring functions inside the cell. Collectively, these structures are all known as the cytoskeleton. And so the cytoskeleton is comprised of different types of proteins, and those proteins are found within the cytoplasm. Okay, so it's the skeleton of the cytoplasm. And although it's not rigid calcium structures like our own bones, these protein structures provide the foundation for shape and anchoring and movement inside the cell. So this is going to be what gives a cell its overall shape and maintains that shape. It's also what's going to allow the cell to change its shape. Okay, and that may happen if you have something like an amoeba that's going to use the movement of its cytoplasm and its plasma membrane as a way to move its entire body. And so we could have cells that are moving using their cytoskeleton components, either interiorly through moving the cytoplasm or in having external cytoskeleton components for swimming behavior. And it also can be used in anchoring. And so we can anchor organelles to structures inside the cell or to each other so that the organelles don't move around within the cytoplasm. And we can also anchor the cell to a neighboring cell, right? So that we can begin to bring cells together and form a tissue. The cytoskeleton components are also essential in cell division. The cell cannot divide without using the cytoskeleton to help separate the chromosomes and also to push the cell apart and stretch it so that it can be pinched into two separate cells. So let's talk about the three different types of cytoskeleton elements. The first is the microfilament, and microfilaments are the smallest of the cytoskeleton components. Okay, they're primarily going to be used for movement, and they're able to be used for movement because of their structure. So they're made up of long strands of protein, and this particular protein is known as actin. And so if you look down here in this image, each one of these little spheres is an individual monomer of actin, so an individual unit of actin. And we have two strands here, one going this way and one coming this way. These two strands are twisted together, okay? That shape, that structure, is going to be important for the function of these microfilaments. If this microfilament wants to get longer, it's just going to chop off some of these monomers. And as it gets rid of them, if it wants to get shorter, it's going to chop off those monomers. And as it gets rid of them, it's going to shrink up, right? If it wants to get longer, it can just add monomers from the solution, and in doing so, lengthen, okay? So this strand is going to be able to get longer and shorter really easily. So if it wants to help push or move things inside the cytoplasm, it can do that by lengthening. If it wants to bring things together and contract, it can do that by shortening. Okay. Microfilaments are really important in cell division. And so here we have a cell that's beginning to divide, and this pinching zone here is called the cleavage furrow. This is where the cell is going to ultimately separate. And that separation is happening because these microfilaments are contracting, kind of like drawing the drawstring on a pair of sweatpants. As they contract, they're going to pull and pinch the cell together. Okay, intermediate filaments, as their name implies, are intermediate in size. These are the middle-sized cytoskeleton components. And if you look at their underlying structure, they are much more permanent. Okay, they're actually made of a very tough rope-like fiber, and that fibrous subunit is twisted with another one really, really tightly, like plying two strands of thread together. And because of that tight wrapping, we can't just make this longer or shorter by lengthening or shortening these subunits. Right? Once those two subunits are twisted together, they're a firm rope-like structure. Okay. So these guys are much more permanent. They don't form and reform. They don't shorten and lengthen. They're they're much more permanent, rigid structures. And because of their rigidity, they're able to resist tension. So when the fluids of the cell push on them, or when neighboring cells and external forces push on the tissue, they're going to be able to resist that tension and therefore help keep the cell firm and keep the body tissue more rigid. The last structure of the cytoskeleton is the microtubule, and microtubules are the largest. Okay, and their size in part is due to the fact that they have a hollow center. And so these are long, hollow tubes. They're cylinders that are produced of tubulin protein. So tubes made of tubulin. And here, each tubulin molecule is two little spheres. So it's a dimer. Instead of a monomer, mono being one, it's di, two. There's two pieces to the subunit. And these subunits are stacked together in a sphere with an open center. So they look very much like a straw. If you turned this image, you could look down through the center of the microtubule. And so these guys can be pretty rigid and are also strong. They're also quite large, and so they can actually function as a framework. And so these end up being the structures inside the cell that provide the building framework and also the transportation network. So they can function both as the rigid internal skeleton that gives the cell really its overall shape, but they also form a network of connected structures, and it's kind of like train tracks that vesicles can move along and motor proteins can walk along in order to move things from one point to another point inside the cell. So this is an image from a video, and I'm going to show you that video in a minute, but I want to tell you what the parts are before I show you the video. So down here, you can see it's pretty large in size. This is our microtubule. And there's another microtubule here in the background. Okay? So these are those structures that are giving the cell its shape, and they're also providing a pathway for proteins to move along. And so this structure here, see if I can circle it, is a motor protein. And that motor protein attaches at this end to the vesicle. That's what we're seeing in the background. And then at this end, this section here and this section here are able to twist back and forth and walk, and they drag this vesicle along this train track. So let me go out and pull up that video. And again, I'll link this video on our Moodle page as well. This is actually just a small excerpt of a much longer video. But here we have all of our microtubule structures giving the cell shape and providing this network inside the cytoplasm. And then here comes a motor protein moving a vesicle along that cytoskeleton. And the motor protein really looks like it's walking. All it's doing is twisting and shifting that bottom section back and forth. And as it does that, it's able to actually drag the vesicle along. Okay. So we also have some important structures outside of the cell that are made up of microtubules. Okay. So remember, microtubules are long and hollow and they're large. And they actually can be formed into even larger structures by repeating rings of those microtubules. And so, two important structures for movement are cilia and flagella. And both the cilia and the flagella are made up of microtubules. And if you look in this image here, each one, sorry, each one of these rings is a microtubule. And so they form in pairs around the outside, and then they have these protein linkers, and then there are microtubules in the inside. And so this cylinder shape is a large protein that makes up the flagella and the cilia of um, various eukaryotic cells. So cilia, that word and its root means eyelash, and flagella means whip. Okay, and so if you look over here, the cilia look like little eyelashes, and the flagella is like a long, narrow whip. Okay, both of them can be used to move entire cells or to move fluid over the surface of a cell. So here is another little paramecium. This is a surface scan of the outside of the paramecium's body. All of these little hair-like structures are cilia. And when we were watching that video of the contractile vacuole, if you looked at the outer ring of that video, you would see all these little hairs beating. And those are the cilia, and they're actually moving water around the body, and they can be used to move the organism through the fluid. They can also be used to create a current and to bring materials to the surface of the animal's body. So, in a single celled organism like this, the cilia are used to drive yourself around in the fluid. We also have cilia in our respiratory system to help prevent debris and dirt and particles that we inhale from getting down into our sensitive lungs. And so we use the cilia to sort of beating those cilia helps to move those things away and to actually absorb them in the mucus of the cells up in the upper respiratory system before you get into the lungs. Um, these guys move with a stiff power stroke and a flexible recovery stroke that looks kind of like what your arms do when you're doing the breaststroke if you're a swimmer. And so they have these sort of many numerous little hairs doing the same repetitive motion. In comparison, when we have a flagella, we usually only have one or a couple, and they're much, much longer. Instead of short little eyelash like hairs, they're these long snake like whips, and they move in a snake like motion. And they're going to be used as sort of a power propeller to move organisms through the water. So there's a lot of single celled organisms like Euglena and some other protists that are going to swim with this tail. And this particular image here is a human sperm cell that's going to use that flagella to swim through the reproductive tract. So this image here sort of shows the power stroke of the cilia and then the return stroke. And they're going to use that motion to move this organism around in its environment. Or in this case, the cilia of the trachea are going to help capture um, particles and debris as they move into the respiratory system, and then they're going to capture them and move them over to these mucus secreting cells where they can absorb those particles and break them down and remove them before they get to the lungs. And then this image is showing the snake-like undulations of the flagella propelling the cell forward by moving fluid away from the direction of motion. Okay, and the last structure made of microtubules that's important for us to think about is centriole. And the centriole has similar structure to the cilia and the flagella in that it's a bunch of microtubules sort of around, arranged in a spindle. And it is also very similar in structure to something that's called the basal body. So in an animal cell, we have a centriole. In a plant cell, we have this basal body. They're very similar in structure, and they're essential for cell division. And so here we have, in black, these are the chromosomes of a cell that's getting ready to divide. And all of these dark lines are micro, um, filaments. They're going to be important for movement, or the mitotic spindle. And then in the center here, one on each pole, we have the centrioles. And so these are the organizing centers for the mitotic spindle, and they're essential, to help prepare all of these structures for moving the chromosomes during cell division. When we come back and talk about mitosis, we'll see this in action. Okay, then I want to talk a little bit about what is on the outside of the cell. And so, even when we have cells in a tissue where they're right next to each other and they're forming a whole sheet, we have, a little bit of space between those two cells. And that space is filled with what we call the extracellular matrix. And the extracellular matrix helps to hold those cells together, and it maintains the shape of the cell itself, and also helps to maintain the structure of the tissue by holding everything together. Okay? Components of the extracellular matrix can also orient, how cells are going to move when new cells, when space needs to be made for new cells, or when cells within the tissue need to move. And they play an important role in chemical signaling. So here we have the plasma membrane. Ah, I'm trying to draw this plasma membrane. And then we have some proteins. They're embedded in the plasma membrane. Down inside the cell, we've got the cytoskeleton. And then outside, we have the extracellular matrix. And the extracellular matrix is usually made up of quite a lot of fibers of collagen. That's what's shown here in yellow. And then also some glycoproteins. And so this is a polysaccharide chain, and it has proteins attached to it. So glycoprotein, a sugar protein complex. And those are going to help adhere cells to each other and, fill in some of this extracellular space. And you can see that they kind of anchor down into these proteins so that they're also in contact with the cytoskeleton on the inside of the cell. Okay, then I want to talk a little bit about how things move between cells, get from one cell to another cell, and how cells are anchored together. So plant cells have an additional complication to their structure, which is the plant cell wall. And so here in this image up top, everything that is pink is cell wall. And then the green area is an opening in that cell wall known as the plasmodesmata. And the majority of movement through the cell wall has to occur at that plasmodesmata, because otherwise the cell wall is this thick, complex of cellulose fibers that are embedded in a protein and polysaccharide matrix, and it's really hard for anything to move through that cell wall. It's a formidable barrier. But the plasmodesmata provides a connection, and you can see it goes from this big cell here out into all of the neighboring cells, and it basically forms a continuous plasma membrane so that all of these cells are directly connected, and they can share materials from one cytoplasm to the other cytoplasm. And here this one is sharing to its neighbor, and then over to its neighbor. And so that means that plant cells can actually send signals really rapidly from one cell to its neighboring cell to the next neighbor because their cytoplasm are directly connected through these plasmodesmata in the cell wall. The plasmodesmata itself, the whole, is lined with plasma membrane. So it's got more membrane lining the channel between the two cells. And these connections provide direct movement of materials from one cell to the next cell. In animal cells, we have three different types of junctions. And the first one is called a tight junction. A tight junction forms a watertight seal. Okay, so this is going to be a very tight, um, close seal between two cells. So in this image, what you're seeing here in brown, this is the plasma membrane of one cell, and this is the plasma membrane of the neighboring cell. And these tight junctions are proteins that go through both cells. And as they go through both cells, they press those plasma membranes together. And so I like to think of this as being like quilting, where each one of these proteins goes from the inside of this cell through the intracellular space. It connects to the next protein, which goes through the rest of the intermembrane space and into the next cell. And so it's going to lock those sections of plasma membrane together, just like quilting holds the fabric. Top fabric and bottom fabric are held together with a space in the middle. And what this does is it means that fluid that comes from this space here, let me turn this back into a pointer. Do that. That's not what I want either. Anyway, for here in this space, right here, and material diffuses out through this plasma membrane and into the intermembrane space here, it can't just go up and around this cell and out to some other cell. Instead, it has to go through and into the neighboring cell because these tight junctions make it so that nothing can go through here. And so this watertight seal also helps to direct materials. So they have to move directionally from one cell to the next cell through tissue. And so they're going to be able to have a cascade or a wave of material movement from cell to cell. The next type of junction is called a desmosome, and desmosomes are anchoring junctions. These are going to be tight areas where we anchor the plasma membrane of one cell to the plasma membrane of another cell. These are kind of like rivets or spot welds. They're a location where the two cells are held together very, very tightly, and they help to form the structure of cells and tissue by kind of anchoring the plasma membranes together, and then also anchoring to intermediate fibers within the cell so that they're connecting from the intermediate fibers of cell number 1 through the anchoring junction protein, across the intermembrane space, and directly to the intermediate fibers of cell number 2. And by having all of these connections, we're basically forming one structural unit. It's going to help keep those cells locked to each other and are going to help to form the integrity of the tissue. And then the last type of junction we have in animal cells are gap or communicating junctions. And gap or communicating junctions are sort of like plasmodesmata for animal cells. They are a protein channel that passes directly from one cytoplasm to the second cytoplasm. So here's our plasma membrane for cell number 1, for cell number 2, and this junction basically forms a tunnel through the plasma membrane so that materials that are over here in cell number 1 can just travel through the tunnel and arrive in cell number 2. Okay? These tunnels that form the junction, these gap or communicating junctions, are able to open and close. And so here we have a closed one. And when it's closed, no material can pass through, right? But when it's open, now materials can pass through the tunnel. And so these can form ports between cells that can regulate the timing of when materials can pass through, and can also help to propagate signals by sending information through that communicating junction. Okay, I'm going to stop this recording here and upload the rest of eukaryotic cells, and I will make a separate recording for prokaryotic cells and plasma membranes, which will take up the rest of our class time for today.